Well, thanks for everyone for coming today. It's a great pleasure to introduce our next speaker for the APGC seminar series, who is Diego Jarquin. Is right? Jarquin is the proper pronunciation, Diego?
Yes.
Yeah. Diego completed his PhD in statistics at the University of Postgraduate Education in Mexico, and after that, he held several positions in the U.S. at the University of Alabama, Birmingham, and at the University of Nebraska in Lincoln, before joining to the agronomy department at the University of Florida as an assistant professor. So Diego merged statistical methodology, AI, computer algorithms development, data science, and collaborative work with plant scientists in his research that is focusing on advanced how prediction models are developed for the selection purpose in plant breeding. So I hope you all guys enjoy the Diego's talk. Diego, it's over to you.
Okay, thank you. I'm going to start sharing my screen. My screen?
Yeah, it's there.
So first of all, thank you so much, Raul, for the invitation. Thank you for the Advanced Center for Advanced Plant Growth Center. It's an honor for me to be here. So when I was initially contacted by Raul, and he mentioned the idea of giving this seminar, he pointed a little bit of, he wanted a more broad set of topics. So I'm gonna try to combine that with the things that we have been done. In advance, I apologize if at some point the connection is not good. I'm currently traveling in Colombia, visiting some of the collaborators that we have here in Ceniparma, and the name of the place is Barranca Bermeja. Okay, I'm going to be talking about plant breeding with genomic selection and high-dimensional predictors. And this is the outline of the presentation. First, I'm going to be talking about what is genomic selection. And this might be something that most of you already know, but we also have people coming from different fields. So I'm going to try to give the details of the concept. And then I'm going to mention about genotype and environment interaction, that is an effect that sometimes generally causes some issues for improving genotypes across environments, and then how we develop prediction models in that case. And then I'm going to talk about a topic that we like a lot, is resource allocations, pretty much sparse tested designs, how we can allow, how we can have a better way to optimize the use of resources. So how many genotypes we should be testing in environments. And then finally, I'm going to talk about Chido. Chido, for those of you who speak Spanish and probably close to Mexico, is a word that we use to refer to something is cool. In our case, we named this software Chido because it's a very easy way to integrate data from multiple omics, and then we can conduct predictions. And then I'm going to present how you can access the platform and a live demo. So first things first, so let's talk about the labor of the breeders. So we are living in a world that keeps growing, right? By 2050, it's expected that the world population is going to be around 10 billion people. One of the biggest challenges of the humanity is to ensure, right, the current and future food supply chains of a growing population. So that's one challenge. The other challenge is that we live in a world that is facing more often and more extreme environmental variations. So you see, this is a bad combination. So it's expected by each degree Celsius that the temperature increases, the yield performance of the elite varieties is going to decrease by 6%. So in this case, the breeders play a main role. Why? Because they have to develop these improved genotypes. And when I say improved, to be more productive, resistant to biotic and abiotic stresses, to be stable, etc. Okay, this is the way how currently, not currently, but in general, the genotypes are being developed. The improved genotypes At the beginning, we have in the breeding pipeline a very large number of unique genotypes that are being tested in few locations. And then the most promising genotypes are being selected and tested in more and more locations. At the end, after a long period of time, a few genotypes are just selected and released as commercial varieties. Obviously, the less genotypes are being selected, the most environments are tested. Why? Because they have to ensure that this genotype is going to be stable across a wide range of environmental conditions. So the interesting part here is the genotype is being released here at the end. It could be between 3 to 5 if we are lucky. The genetics of that set of genotypes is the same that we have right at the beginning. So if we are able to find a way to identify those genotypes at very early stages, we can save a lot of time. In general, with annual crops, this process might take between 7 to 8 years. Just imagine with perennial crops, it's way more complicated. So some of the inconveniences of this conventional breeding scheme are that we have, in general, limited resources. We don't have enough seed. The ideal would be test all genotypes in all environments, but we don't have enough money to pay for the experiments. The seed is not enough. We don't have land, water, and it's very expensive in general, phenotyping costs. And the current the pipeline is time consuming. As I said before, it might take up to 8 years developing these improved varieties. However, in our side, we have the technological advancement that can help to alleviate these issues. So we have improved sequencing technologies. These modern methods can help us to characterize genotypes with high detail. We have envirotyping. We can characterize in the same way environments collecting weather information. And also we have high-throughput phenotyping methods where we can have a good characterization of the phenotypes in fields. And also we have improved prediction models. We have parametric methods, penalized, Bayesian, non-parametric, artificial intelligence, machine learning, deep learning, random forest, all these new methods, and hybrid methods that combine both things, right, via kernels. So dealing with complex traits, right, the economical traits in general are complex in the sense that they are affected by a large number of small gene effects. And I think what is crucial here is that these gene effects are small, and these are affected or modulated by the environmental conditions the genotypes experience in fields. In a way, the genes and environmental factors interact in many complex ways. So one of the challenges is to cope with highly dimensional network genomic data and environmental information, but more important, how to grab all this information in a way that we can make sense to the interactions. The number of interactions can be very large. So in our favor, we have these new methods like whole genomic prediction that can help us to reduce the selection time, right? Initially, the concept is very easy to understand, but very complicated to apply in the real life. So initially, we require a set of genotypes for which we will have phenotypic data and also molecular marker information. As I said before, phenotyping the genotypes in fields is very expensive. Sequence those genotypes is becoming cheaper and cheaper. And then we have a model where we can combine these two sources of information and we train the model. So the idea is once we train the model, we can test the performance of the test candidates, right? The potential candidates for selection via their marker profiles only. In a way that we can provide a set of predicted values, surrogates of observed values to the breeders, and they can use that information to select the improved genotypes. In this case, we are not interested in selecting specific regions of the genome, like QTL mapping of major genes, so we don't test for significant markers. So in general, we don't apply genomic selection right away in the real life. We have to conduct some tests, some experiments to see with data that we already observed to evaluate whether the data that we have at hand is good enough to conduct the predictions. So for that, we have to consider a cross-validation scheme where we use a specific set of data or data points as training for the model and as the remaining data points as testing for the model. The idea is to calibrate the model to predict the unobserved genotypes. In this case, we are mimicking we didn't observe those genotypes. And then once we predict those, we can use compute the correlation between predicted and observed values, and that will be a method that sometimes we use to evaluate whether the model is going to be good or not. Okay, this is the basic idea. However, depending on the stage of the breeding pipeline, we can consider different cross-validation schemes. So we develop or we propose 4 different cross-validation schemes that can be applied for different stages. So CV2 is what we call incomplete field trials. And here we are trying to predict genotypes that we already observed in environments where we didn't observe those genotypes. So in this study example, we have 5 lines tested in 5 environments, but line 5, for example, was not observed in environment 1. We have plenty information of this genotype in other environments and also information from other genotypes in the same environment. Usually we conduct a K-fold design where we randomly assign phenotypes to folds. The second one is cross-validation one, where we are predicting untested genotypes in observed environments. Here, we assume that we didn't observe that genotype in any of the environments, and we use the information from other genotypes in those environments. Let's say that this was the initial idea of how genomic selection was proposed, right? The idea was to predict a set of genotypes that were not observed at all. Here we have a K-fold design where we make sure that the same phenotypic records of each genotype are located in the same fold to avoid contamination. The next one is CV0, where we are predicting genotypes in environments that we haven't observed yet. So here we are interested in predicting genotypes in environment 3. So we discard all this phenotypic information, and then here we are leaving out one environment. And obviously, the most difficult and most interesting prediction problems for breeders is to predict untested genotypes in unobserved environments. Here, our target is to predict genotype 3 in environment 3. So for that, we have to discard all that information for that environment and all the information of that genotype across environment. So here, basically, we are sort of leaving one environment out and one genotype out at a time. Obviously, at the end, it's not like that because it's very time-consuming. So here I summarize using colors the different cross-validation schemes. So in CV2, we randomly assign 5 different colors to each one of the genotypes. We have in the columns the environments, in the row we have the genotypes. In a way that combinations with the same color are assigned to the same fold. In the bottom left, we have cross-validation 1. Here, all the phenotypic records from the same genotype are assigned to the same fold. And we have CV0, where we are discarding that environment, and also we are predicting use a specific fold in that environment. And finally, CV00, we are discarding information of that environment, and we are predicting one specific fold in that environment, but we take extra care to eliminate the information from the genotypes that we are predicting, but those were observed in other environments. So we make sure that we are avoiding contamination. That's it. Later on, we evolved these methods to consider perennial crops, where we have genotypes that have been observed in some environments across different years. So year 1 for genotype 1, year 2 for genotype 1, and then for the sites pretty much. And then we can combine or we can craft the previous cross-validation schemes. And also another important prediction framework is forward prediction, where we are using information from 1 year to predict the performance in the next years of the genotypes. There are some crops, for example, where the first year are used for establishment, but some of this information can be useful to predict the performance in 2 or 3 years ahead. And also we can use information from 1 single year to predict the next one, or combine year 1 and year 2 to predict the next year. Regarding the metrics, we use the person-moment correlation between predicted and observed values to evaluate the ability of the model. In this case, we are working with continuous information, and with traits like yield and high disease resistance, we care about the rankings pretty much. So we rank the genotypes and we are selecting in one of the tails. If we are considering only one environment, we use the Pearson correlation. If we use multiple environments, we need to account for the user title of the predictions at given environments and the sample sizes. So we have to calibrate, right? We have to pool and give a specific importance to the different environments. So we use a formula like this to get a weighted average. Okay, now I'm going to talk about genotype by environment interaction. As I said before, breeders are interested in developing genotypes that perform other genotypes in a wide range of environmental conditions or environments. For that reason, they have to establish trials in multiple environments. However, it might be the case that in some of these environments, we have a specific performance of the set of genotypes, and the negative environmental conditions might be the case that one of these genotypes is the only one that can make it, right? So it might be the case that we can discover a genotype that is resistant and the others won't make it under these negative environmental conditions. So in that sense, we have the G/E map. So what is the G/E at the end? So in the broad sense, it's a change on the rankings of the performance from one environmental condition to another environmental condition. In this case, we have three genotypes. Genotype B has a low performance in a low environmental value. It could be temperature or it could be an index. High performance in a high environmental value. Genotype C has an opposite behavior, and genotype A is stable. So as I said before, the breeders want to select the most successful genotypes across environments. Under this condition, it's difficult to pick a unique genotype. So because A is stable, but it's not the best at all. So when we have this change in the ranking from one environment to another, we said that we have presence of G/E interaction. So here we can ignore the fact that we have G/E interaction, and we can just select based on means across environments. We can reduce it by ignoring that we are selecting for stable genotypes and just selecting genotypes with local adaptation, or we can exploit this fact by considering the borrowing information across environments. So, and this is what we try to do. We are trying to exploit this this problem. So the current approaches to confront these challenges are the use of genetic information. So we can characterize genotypes using molecular marker information or pedigree. Also, we can characterize environments using weather information, and the interactions that are arising between these two components. So the standard genome-enabled prediction models, they explain the phenotypic response as a function of a genetic signal plus an error term. In general, in cases like this, P, the number of molecular markers, is way larger than N, the number of genotypes. For example, we have some cases, we have 100 genotypes and 1 million markers. So we have some limitations in that case. Also, we have this model, the standard model does not include weather information, neither the interaction between the environmental covariates and the markers. So we can divide pretty much the approaches into big branches. One branch where we have genome-wide association studies, where we are considering the selection of molecular markers with a high signal. On the other side, whole genome regression and prediction, where we regress all genotypes at once. So let's start with a simple model. Let's say that we are trying to explain the performance of the I genotype as a linear combination between marker and marker effects. As I said before, P is usually greater than N. And that poses extra challenges. Why? Because we will see later it's difficult to get a unique solution. We have more predictors than data points to solve the equation. So in that case, we have to use a different set of models different to the ordinary least squares. We have penalized methods, where we are adding a specific shrinkage or penalty to the models in order to be able to get a solution, but that reduces the chances to select or reduce the number of covariates that we can select. On the other hand, we have the Bayesian approach, where these pretty much are the Bayesian versions of the penalized methods, where we are adding a prior density, and this is going to allow us to select or use as much as the genomic information is possible. So let's talk about how we can extend these standard prediction models that are for allowing the high-dimensional markers, high-dimensional covariates, and interaction between them. So let's start with a very simple approach, with a simple concept, I would say, and I promise I'm going to talk very few about statistics. So first, let's review the effect of shrinkage on the mean square error. So the mean square error is defined as the difference between the predicted and observed value, the square of that, and that can be decomposed as the sum between the variance plus the square of the bias. If we have an estimate of this bias, we multiply bias, that estimator by a value between 0 and 1, we have this new estimate of theta tilde is equal to alpha times theta, where the variance of this new estimator is equal to alpha squared, the variance of theta hat, and if alpha is less than 1 and greater than 0, we have that variance is going to be less or smaller than the original variance of theta. So in that case, we are adding a bias into the solution in order to reduce the variability. So we can have more precise estimations, but we are inducing a bias in that case. So this is the relation that we have between shrinkage and the penalized methods. Let's say that we start again with the model that we described before, linear combination between marker and marker effects. And then we have two methods for conducting the estimation. We have penalized regressions, that is that trade-off between model model goodness of fits and model complexity, where we are adding a penalty, right, to the model. We are in this condition because we want to be able to get a solution for each one of the effects of the markers that we are estimating here. So the solution to this model is equal to beta hat x prime x plus lambda, the inverse of that, x prime y minus 1. That's what we call rich regression block. And if we have the Bayesian regression, we just have to add prior density. So the posterior density is proportional to the likelihood times the prior density. We assume that the marker effects follow a normal distribution. So the important part here is the posterior mode is equal to x prime x plus the ratio between the variance components of the error and the variance components of the marker effects that we specify here, times the inverse of that times x prime y minus 1. And if you pay attention, these two solutions are quite similar. On one side we have lambda, on the other side we have the ratio of the variance components. So pretty much this lambda can be viewed as the ratio between the noise and the signal, right? If this ratio is large, we are inducing a large penalty in the model. So in this case, we are using this trick to break the singularity. Otherwise, we won't be able to get the solution to this, the inverse to this matrix x prime x. Remember, this is of order p by p. We have only n data points to get a solution. So we have complexities dealing with high-dimensional covariates matrices. So in this case, still we are able to get a solution, but use in mind, if we have 100 genotypes, 1 million markers, the size of the order of this metric that we want to invert is the order 1 million by 1 million, although we only have 100 genotypes or data points. So that's why we start, now we review this star model that was originally presented in animal science, what we call U-block model, that here we following the same condition where beta follows a normal density centered in 0 sigma square beta. Instead of being interested in estimating the marker effects, we are interested in estimate the whole linear combination between markers and the marker effects. In this case, is the genomic effect of the ID individual, and this can be viewed as the product between the vector of markers times the vector of effects. If we stock all of the genomic effects in one single vector, we have that this vector is equal to the product between the X matrix, the matrix of markers, times beta. And the covariance of this G is equal to the covariance of X beta equal to X prime sigma square beta. So we have that this new vector of genomic effects follows a normal density, XX prime, so what we call G matrix or kinship matrix. So in this case, G is the kinship matrix whose entries describe genomic similarities between pairs of individuals. So in this case, we have that the solution for this new model is equal to I plus lambda G inverse Y minus mu. In this case, this lambda, as we said before, is the ratio of the variance components. Here, the advantage of getting this solution is that now we are dealing with matrices ordered n by n. So if we originally had 100 individuals, when we get the solution, after inverting this matrix, that this matrix is of order 100 by 1. So we are reducing the dimensionality by a lot, and the solutions at the end are the same. So we are getting exactly the same solutions in different ways. Either we estimate the marker effects, or if we obtain directly the linear combination of the markers and marker effects. So those two methods are equivalent. However, this is computationally speaking more convenient. Okay, now we mentioned very quickly how we can integrate genomic data with high dimension in the models in an easy way. Now let's see how we can integrate environmental data in the model. So the environments can be characterized with also a large number of environmental covariates. So that can be measured in a continuous, for example, daily basis, hourly basis, every 5 minutes, 5 seconds, etc. So we have a very large number of covariates there. Suppose that we have a model, right, where we are now trying to integrate genomic data plus environmental data. So we use the same trick, right? Instead of characterizing genotypes using genomic data, now we are going to characterize environments using environmental information. And we use the same assumptions. So in this case, the environmental effect is a linear combination between environmental covariates and the corresponding effects. In this case, we also assume that the corresponding effects follows a normal distribution, centered on 0, and variance sigma squared sub gamma. So after we repeat the same derivation than what we did for the G-block model, we end up with the case that W, the vector of environmental effects. The covariance structure of this vector is equal to W prime sigma squared sub gamma is equal to omega. So in this case, this omega can be viewed as the environmental kinship matrix, where the entries describe environmental similarities between pairs of environments. So if we put together these two developments, these two derivations, we have with this new model, a common effect plus environmental effect plus a genomic effect with the following conditions, right? G follows a normal density, centered on 0, and variance covariance given by G. W, the same, centered on 0, and variance covariance given by omega. And again, as I said before, this omega matrix describes similarities between pairs of environments. So it allows the borrowing information across environments. If we have environments that are similar to other environments, we might expect that the borrowing information is going to be higher in that case. So the limitation of this model is that it does not allow the inclusion of interactions. So let's now learn how we can incorporate interactions in this model. And a way to include this interaction is how we learn the whole stats courses, right? Via contrast. If we have two different set of covariates, P markers and Q environmental covariates, the number of contrasts that we can construct with these two set of variables depends on the dimensions. For example, we have two markers and one environmental covariate. The number of contrasts of product between these two variables is 2. If we have two markers and two environmental covariates, the number of contrasts is 4. So it sounds interesting, but remember, we already had issues before trying to add a large genomic information in high dimensions. So considering these interactions, it can be very complicated. So the number of contrasts can grow drastically. Typically, in this type of studies, we have between 50,000, 500,000 markers, and the number of environmental covariates can be large. Let's say a toy example, we have 2,000 markers and 100 environmental covariates. The number of contrasts that we need to add in the model is 200,000. So this is quite a lot. Then we start thinking, how can we integrate these interactions in a similar way that was done with the G-block model? Let's say that we have two scores, genomic scores, that is going to be equal to the linear combination between marker and marker effects, with the marker effects following ID distributions. In this case, those are not normal. We are not specifying a specific distribution. And we have something similar to the environmental effects. W sub I tilde follows is equal to a linear combination between markers, sorry, between environmental covariates and the corresponding effects, with effects following ID densities. Now we assume that the marker effects and the environmental effects are independent. We know that this is not the case, but we are just proposing the model in a way that we want to evaluate if it works. Also, we go beyond and we also specify the expected value of this linear combination is going to be centered in 0 because beta is equal centered in 0, and for W sub tilde the same. And the covariance between the II entry is proportional to the I prime entry of the G matrix, the kinship matrix, and the covariance between I prime record is proportional to the I prime entry of the parameter kinship matrix. So if we propose a multiplicative model, it's going to be of this form. G W sub I is equal to the product between G sub I times W sub I. So in order to specify a prior density for this term, we need to know two things, the mean and the covariance function. So we derive this. GW sub I is equal to G sub I times W sub I, so we can separate this. The expected value of this is equal to the expected value of the terms. And we already specified before that the expected value of each one of these terms is equal to 0, so expected value is equal to 0. The covariance between GW sub I and GW sub I prime record is equal to, by definition, the expected value of the product minus the product of the expected values. We already showed that this is equal to 0, so we focus on this part only. So we can separate the expected value in this case. We can separate the terms. It's equal to the expected value with G sub I, W sub I, G sub I prime, and W sub I prime. And we can aggregate it. So we can put together the G's and together the W's. And this is equal to the covariance of I prime record in the kinship matrix and the I prime record on the environmental kinship matrix. So pretty much we are multiplying the corresponding cells on each one of the terms of the corresponding covariance structures. So when we have this case, the cell-by-cell product is what we call Hadamard product, and it's specified by hashtag. Right? In this case, this new model term allows the borrow information only between environment by line combinations. They are similar at the level of the genetics and similar at the level of the environment. So if these two things happen, we might ensure that the borrowed information is going to be good. Otherwise, if one of these has a low value, the borrowed information is not going to be that significant. And this has linked with previous literature. From 1954, Cockerham and Kempton, they proposed this way to integrate covariance structures for study the degree of resemblance between relatives by epistatic interaction. So now we have a model that includes common effect, environmental effect, main effect of the markers, and the interaction between markers and environmental covariates. So pretty much conceptually, we are including the interaction between each marker and each environmental covariate that we have. And these are the assumptions. W follows the normal density, centered in 0, and a covariance structure given by omega. G, something similar, centered in 0, and covariance structure given by G. G sub W, normal density, centered in 0, and various covariance given by G as a mark product omega. So sometimes it's important to account for imperfect information and model specification. So if we already have a model where we were able to include the interactions, it might be the case that we have imperfect linkage disequilibrium in the data, marker data, or that the current environmental covariates are not enough to characterize the environments. So in that case, it's needed to bring information that is related with these two terms. We have, for example, the effect of the genotypes associated with the markers and the effect of the environment associated to the environmental covariates. In a way that we can have an extended model where we are including these main effects, environment effect, line effect, and also the interaction between markers and environments. So here we have the interaction between markers and environmental covariates, and here between markers and environments. So now I'm going to talk about the data that we use to test the model. We have a data set. It's from 139 with lines tested at 340 sites in France for a total of 7,876 phenotypic records. So of the total potential cells, only 60% were observed, and only one line was tested across all environments. This is how the allocation looks like. We have in the x-axis the environments, in the y-axis the genotypes. Each vertical line represents a combination of genotype in environment that was observed. So the area in this case, in black color, should be 60%. 16%. So here we have 3,548 markers, and after applying quality control, we end up with 2,395, and we use conventional quality control. We discard those markers with more than, with manual frequency greater than less than 3%, and percent of missing values greater than 10. And environmental information, we have 130 covariates, and after applying quality control, we end up with 68. This is the sequence of models that we consider. We have models that include only main effects and models that also include interactions. So we have 4 models with main effects. So we have a model without marker information, without weather information, and then the models that also include interaction between markers and environment, markers and environmental covariates, and both type of interaction. So also we consider the estimation of the variance components. And this is an important measure because it can help us to evaluate the importance of the different model terms. So here we have a toy example where we are explaining the performance as a sum with a common effect plus a random effect of the genomic information plus an environmental effect based on weather covariates. So here, if we compute the expected value is going to be used as a constant, right? Because all these are centered in 0, and the variance is equal to the variance of assuming the G and W are dependent is equal to the sum of the variances. So it's equal to sigma square sub G plus sigma square sub W plus sigma square sub D. So in this case, we can evaluate the contribution to the to explain variability of each one of the terms as the ratio between the corresponding factor divided by the total variance times 100. So this is some sort of heritability, for example, right? We can identify what's the percentage of the contribution of each one of these model terms. Okay, now the empirical assessment was done considering two different cross-validation schemes. So here, as I already mentioned this, we assume that we are breaking new genotypes observed in tested environments. We consider a tenfold design, and the one is in complete field trials where we have genotypes that were not observed in some of the environments. And these are the results.
Diego, sorry. Diego, sorry. Can you close the two windows that appear there?
The phenotype was close to a normal density. And then also we check the variability of the genotypes across environments and the variability across lines. So it's very clear that we have more of the variability is addressed by the environmental. These are the results. Here we have the variance components. With the simplest model, we have 200 units for the environmental factor. And as we keep adding the information in the model, we started reducing that variability. And also we reduce the residual variance from 22.6 to 14.9. Since we cannot do much for explaining the variability addressed by the environment, so we consider the within environment variability. So after discarding this component, we start evaluating what the contribution of the different components. So now the line effect has a contribution of 35.2% and residual variance 64.8. Here, when we keep adding more terms in the model, we were able to reduce the residual variance from 64 to 26.1. So here we have interactions capture around 15% of the variability. So it's important to include those interactions in the model pretty much. So we have here the result for the default cross-validation. We have the model that includes only main effects of the markers have a performance around 0.19. In this case, it's the G-block model, but we would have gotten similar results using any other of the main effects models. And then as we keep adding the interaction, we were able to improve predictability until 0.23. That represents an improvement of 20% with respect to the main effects model. Another way to evaluate how good or how bad the model is performing is based on the conditional plots. Here we have in the x-axis the predicted values for different quantiles, 20, 50, and 80%, and the y-axis the observed values. If we select the top 20% of the predicted values, we have around 63% has a performance above the mean fields. In this case, for CV1, predicting new genotypes. For CV2, as expected, once we already observe the genotype somewhere, the marker information loses relevance. Right now, we have the same result using marker information or without marker information. However, when we consider the interactions, we observe an improvement of around 20%. And when we look at the conditional plots, if we select 20% of the top 20%, we have the around 80% as a performance above the mean. So in general, it seems like the method is doing a good job. Try to screen the top ones. And then after that, we apply these to other studies. For example, using all the data sets, using data proximity, we were able to improve predictability using marker information or also pedigree information between 42 and 49% with respect to the main effects models for CV1 and for CV2 between 40 and 30%. So one of the nice things is that if we don't have marker information, still we can use pedigree data because it's based on covariates structures. And then another study using 1,000 baselines tested in 5 environments, we observed an improvement of around 143% for CV1 and 147 for CV2. Also, other colleagues have published manuscripts where they consider populations from cotton tested in 9 environments and 78 covariates, and they were able to improve predictability between 166% and 150%. So to wrap up this section, we have we propose a new method, right, to make use of increasing and rich complex data with genomic information, high dimensional SNPs, but also environmental information can be highly dimensional. We have automated systems, we have environments, we have several factors that can be collected, radiation, temperature, rain, as I said before, in a continuous basis. And this method has applications not only for plant and animal studies, also in public health applications where we have problems like personalized medicine, where we have similar elements. We have thousands of the SNPs, environmental factors, social factors, etc., and other type of data that can be included in interactions in the models. So we offer an approach to deal with this complexity of modern data. Okay, now talk about how we were able to leverage the knowledge that we generate with this previous set of models. So one of these is the resource allocation, where we have sparse testing designs. So remember this image corresponds to the conventional breeding pipeline at the industrial level, where we have a large number of genotypes that are tested in few locations, and then the most promising have been selected to be released as commercial genotypes, right? So as I said before, the idea is to identify the promising genotypes at very early stages to reduce time and resources. As I mentioned, the ideal will be to test all genotypes in all environments. So we have 150,000, we have 100 environments, but also I said there are a lot of restrictions, right? We don't have enough resources, availability of seed, etc., etc. But if we analyze a little bit what the percent, how many phenotypes are being observed across the whole breeding pipeline, assuming that the environments are exchangeable, meaning how the 100 environments that we have here at the bottom, we have that the same environments are the ones that we have at the top. We know that this is not the case because those change across the time, but let's assume that. So in that case, after making the numbers, the total number of phenotypes that we collect during the breeding pipeline is 1,850,000. So corresponds to 12.3% of all of the potential combinations. So if we can leverage these resources in a way that we can pretty much be able to evaluate all genotypes in all environments, that somehow we can save resources for sure, and more important, the time. So in these cases where we start working with the sparse testing designs. As I said before, the breeders are interested in release these improved varieties, and they should be stable across environments and should be better than the current elite materials. So they have to conduct these multi-environment trials, and as usually happens, we don't have enough budget to test everything everywhere. But sometimes there is budget for a reduced number of combinations of genotypes in environments. In the previous scheme, across the 7-8 years, 12.3% of all the combinations were observed somehow. So assuming the same cost for testing plots across environments, the question to consider are, and this is a big assumption because we know the cost of phenotyping across different locations varies a lot. But let's say this is an assumption. So the questions to answer is how many and which genotypes will be observed in different environments, whether we should consider overlapping or non-overlapping genotypes across environments. And this is a question that was asked many times. What is better, observe more genotypes or observe the same genotypes across environments? And also how much we can reduce the training set sizes without affecting the predictability. So I'm going to present results on 4 different species. So maize, wheat, soybean, and sugarcane, different sizes. We have experiment size of around less than 1,000, around 1,000, around 2,000, and this one is 850,000 phenotypic records. This belongs to a private company. And finally, sugarcane with very few genotypes. 189 genotypes tested in 6 environments. So how we develop this concept? So suppose that we're interested in evaluate the performance of 843 genotypes in 3 environments. It means the total number of phenotypic records that we are collecting is 2,529. And let's say that we can test only 1/3 of these combinations, pretty much 843. So the options to split these genotypes in fields is, let's say that we are trying evenly split with 281 genotypes per environment. The question is how we are going to assign those to the environments. We have initially 3 options. Option number 1 in the left, we observe each genotype only once across environments. So the columns correspond to the environments, the rows to the genotypes. In a way, that each genotype is observed only once. And the testing set corresponds to the blank or to the white squares. Option number 2 is the other extreme. We just pick randomly 281 genotypes, and these are observed across environments. And again, the testing set is going to be those in white color. So in both cases, the testing set size is the same. And another option is to observe a set of genotypes being unique across environments and a set of genotypes being common across environments. In a way that we are observing the same number of genotypes within each one of the environments. Here we have 281 in environment 1. Of these, 241 are unique across environments, and we have 40 that are common across them. Also, we have other options. We can reduce the training set size. Instead of having 281, we have 211 with an environment, and then the testing set is exactly the same that we have in both. Or combinations, we reduce to 211, but we have 171 being unique and 40 being common across environments. As I said before, in all the cases, the prediction set size is fixed. So in both cases, on the 3 cases, is 562. And these are the combinations that we tested. We tested different training set sizes from 281 to 141, and the composition. We have those cases where the genotypes were unique, so we observe all genotypes only once, or where we have the same set of genotypes observed across environments. We consider 3 models, the models that I presented before, and this corresponds to the model without marker information as main effects, and the interactions. I'm going to present the results. So first, with the largest training set size, in this case 281. In the left side, we had a case where each genotype was observed only once across environments. In the right side, a set of 280 genotypes was common across all of the environments. So we have the 2 extremes. As we move from the left to the right, we are adding a set of 10 genotypes to be common across environments. In black, we have the results for the model without marker information, and as expected, as we increase the number of common genotypes, we are predicting unobserved genotypes at random. That's why the correlation is 0 at the end. With the main effects models in blue, we have as we keep increasing the number of common genotypes across environments, we start improving predictability. And with the GBI model, we have something similar. But in this case, we already have a performance that is beyond twice the predictability of the main effect model. And is less affected by the training composition. So it's true that we are improving predictability a little bit, but it's less the effect of the training set size in this case. So now let's see what happens when we reduce the training set size. So as we move from the left to the right, we are reducing the training set size by a set of training of 30 genotypes. So 281 to 51 until the end 141. So pretty much we can use half of the training set size to conduct the predictions, and still we're going to have good results. Also, as we increase the common number of genotypes across environments, we keep improving predictability. And very quickly, I will go through all of the different data sets. So we have something similar. The GBI model is the best in this case. The improvements are not that significant with respect to the main effect models. And for the different training set sizes, we also keep, we can reduce the training set size by half, and still we can have good results. For weak data sets, we have something similar. We can reduce, obviously we decrease a little bit predictability, but in all of the cases, the GBI model was the best. For this data set, as we increase the number of common genotypes, we decrease predictability. So at the end, it depends on the different data sets, right? But in general, the main effect model was not as good as the GBI model. So we have another where we try to increase, another data set, we try to increase the number of genotypes in environments. So here we have 1,755 tested in 9 environments. And we have a similar arrangement, 195 genotypes initially, and at the end we have 95. So we vary the training set size between 5 to 11%. And this is the genetic diversity. Here we observe all genotypes once. Here we observe the genotypes, very few genotypes across environments. For 11%, 8%, and 5% for training the model. And these were the results. So here we eliminate the model without marker information, and then we add a model that also includes the interaction between families and environments. So this is a soybean data set comprised of 40 biparental populations, 140 genotypes per population. And the idea was to use information of partial families observed in different environments. And it seems like we can leverage that information very good to improve predictability. And finally, I'm going to talk about this, the case of where we have a large data set, what we call the company data set. We have 2,500 genotypes tested in 340 environments, or genotypes in all environments. The total number of combinations is 850,000 data points. We have information on 2,300 markers. And here I'm going to present results using 2% of all these combinations, pretty much 17,000, for training the model to predict the remaining 833,000. So we consider different ways to compose the training set size. So one is selecting 50 genotypes in a way that we have 43 genotypes being common between neighbor environments and 7 being unique within environments, across environments. Another was to use 50 genotypes selected randomly and observe it across all of the environments. And another was to consider genetic diversity into the picture by selecting cores where we can increase genetic diversity or decrease genetic diversity on a set of 50 genotypes only. We implement what we call supersaturated design. And these are the results. In the x-axis, we have the results for the correlations between predicted and observed values for each environment. For the model where we have main effects only. In the y-axis, the model that also includes the GMA interaction. So we have 2 reps, and we have the average correlation with the G-block model was 0.23, and with the reaction norm of the GBI model 0.57. So as you can see here, in all of the cases, the points were above the orange line, meaning that the GBI model was better than the main effect model. What happened when we select 50 genotypes randomly and we observe those across environments? We improve predictability. So we boost predictability of G-block to 0.3 practically, and with the reaction norm model to 0.6 in average. Just remember, we are using 2%, 17,000 data points to predict the remaining 98,000. Sorry, 98%, 833,000. And when we put together the ideas of the using genetic diversity, sadly, we didn't observe any significant improvement. So we observed that correlation when we increase genetic diversity, G-block model is 0.23, and the reaction norm is 57, and we have exactly the same results when we decrease genetic diversity. And in any case, the message is when you have a very large number of genotypes and a training set, pretty much you can reduce phenotyping costs quite a lot in this case. And finally, we have a data set where we consider sugarcane with some collaborators in Colombia. And here we assign 36 genotypes, 31 genotypes per environment. And the idea was to predict the remaining genotypes in that environment. So here with the main effect model, we got a correlation around 0.58, 0.6 with the GBI model. The interesting part here is that we can reduce training set size by 60% or 66%, and still we can get a good results predicting the observed genotypes. So we have 0.51 and 0.51 with the main effect models and the GBI interaction. So there were not big improvements by adding the interaction component, but still we can show the resource allocation can be reduced. So the conclusions are we have different patterns for different crops and models with maize, wheat, and soybean. So some of these cases, some were stable. In other cases, we reduced predictability by increasing the number of common genotypes, or we increased predictability by increasing the number of common genotypes. In any of the cases, the GBI model significantly outperformed the main effect models when we predict correlated environments. And GBI model returned the best results for all training sets and compositions, and it was less affected by the composition of the training set. It's important because as I said before, with the main effects, in general, you can improve predictability. However, realistically, the breeders do not have enough seed to observe all genotypes in all of the environments. So with this, we can achieve an efficient use of resources, seed availability, land, water, without highly affecting the precision. Okay, now very quickly, I want to talk about the last topic, and it's Chido. So what's Chido? Chido is a no-code application that aims to remove technical and financial barriers associated with modeling genotype-by-environment interaction in the prediction context. It stands for Characterization and Integration of Tribenomics. And the last year I was lucky that I had two very smart students. So one is Francisco Gonzalez. This guy, during the night, he is master student. During the day, he is a model apps lead for North America in Google. So he should know something about programming for sure. And the other guy was a visiting student from Spain, Julian Garcia Badillo, who just finished as intern at Google. So we met the three of us and we started developing this idea of having something that people can get access and use for free everywhere, in a way that they don't have to spend a lot of resources trying to hire a statistician, a modeler, etc., etc. And they can, the breeders or emerging breeding programs, they can have an idea of what the potential of their data. So how can I access Chido? We have two ways. We have on demand, and we can also use the local computers. So this is the link for accessing on demand, and this is the link for the local to download the software in your local computer. So these are how it works. So we have different interfaces where we can conduct different tasks. So we have an interface where we upload the data. We specify some information about the genotypes. And this is the information how we can download in our local computer. We have to go to the GitHub website. We clone the computer or download the zip. We extract the the information, and then we can use this. I'm going to show these instructions for running the demo. So the important thing here is that you have to run the demo when you are inside of this folder where you extract the information. So here we have the demo. This is second, and let's access the platform, right? And then run the demo. I'm going to show you in a second. So you can run the demo, run the implementation using RStudio or using our standalone version. So we have an interface to upload the omics, another to create the models, another to train the models, and another to view the results. So I'm gonna very quickly try to load the implementation in R. Oh, it's here. Second. Okay. Well, now and this is the advantage of using your local computer is that you don't have limitations on the amount of resources those that you have in your computer. So first we browse for the data. We have this data set. Why? This corresponds to 99 double haploids that were observed in 4 environments, and this is how the data looks like. We have to identify the important parts here. We have environments, ID of the genotypes, the trait that we want to analyze, and the unique identifier of genotype in environment in case you have high-throughput phenotyping or data collected at the plot level. So the ID of the genotypes is in column number 5. Environment ID is in column number 1. In column number 12, we have the unique ID, and the target is in column number 6. So these are the columns that we are interested in. This is Grand Hill, for example, and then we upload the data, and we have 2 omics to play with. The next step is to use marker information, and this is how the marker information look like. We have in the first column the ID of the genotypes, and then after that we have the markers. By default, we call this G, and then we upload. And then let's say that we want to add phenomics data. So this is how the phenomics data looks like. We have data that was collected with remote sensing, proximal sensing. We have 62 bands collected in 5 flights, and the first column has information of the genotype in environment combination. And this is important. It doesn't matter if it's not ordered. Internally, the software is going to order this. So let's go high-throughput phenotypic data. Omics. And then the linkage is component ID. We can visualize the information needed. Here we have lines, we have markers, etc. Anyway, let's create the models. And this is the fancy thing that I like. We can just drag and drop. We add the environment, the main effect of the markers, and then we can save this model. We can add interactions with drag and drop, building on the previous model, and we drag and drop 2 terms, so G by E. And then we can save, or we can type the formula directly here. And let's say that we have another model where we add the main effect of the bytes. Save. So here we validate the model. Let's say that we are interested only in 500 iterations, 100 for burning. We set the seed. We consider 5-fold for cross-validation. We standardize and send in the genomic data. And let's say that we run only 2 different cross-validations. So we can run 1 model at a time. Unfortunately, we cannot run this in parallel. And then what it does is start following a set of steps to create the different covariance structures and then perform the group validation. So all of the steps that we have for the prediction pipelines are condensed here. And then we calculate the metrics. As I said before, we have the correlation between predicted and observed value within environments, the mean square error, and the variance components. And then we run one model. Let's say we run the second model. So obviously, when we have one more term, it's going to take a little more time. Then again, we compute the covariance structures and we perform the cross validation. Remember, we have 5 poles, so we are repeating everything 5 times for the 2 group validations. Then finally, I'm going to run the analysis for the model that also includes the high-throughput phenotype. Okay, while it's running, I'm going to mention the next steps with this software is to add other type of strategies for prediction, like AI methods, and also the sparse testing designs and the prediction of hybrids using genomic information from the parental image, or the parents. Okay, it's going to be done soon. So once it's done, so what I suggest to everybody is just to go and download the results right away. So before you lose, I mean, you are accessing this on demand. It might take, you can lose the results when the session is done. If you are running this in your computer, it doesn't matter. And then we can visualize the results, the correlation between predicted and observed values within environments. So we have in the first row, the results for population 2, the row at the bottom, population 1, and the columns correspond to the different models. The last column within each subplot corresponds to the weighted average. So here the correlation, the highest, the best. And here is clear that using high-throughput phenotypic data is way better than using just marker or GY interactions. We also compute the mean square error, the lowest the best for each environment and across environments. And finally, we observe the variance components. So remember, this gives us an idea of what's the most, the factor that contribute the most to variability. Also, you can see the same results in the zip file that you download. Okay, this is sort of going over the time. This is something I wanted to show you. So I have to thank my people in my lab. These are some of the current members and members who are helping to develop things. So with this, Gogators, and thank you for the time, and I will be more than happy to answer any questions.
Thanks, Diego. Thanks a lot. Thanks for the presentation. Very interesting topic. There is several questions in the chat, so I would like to, if someone have question in the chat, want to unmute himself and ask the questions. If not, I will read it. So, for example, Catherine, you have a question at the beginning?
If your Bayesian regression was to assume distribution and the variance in the prior, because I'm kind of wondering how much the, well, linkage disequilibriums disrupting normality would affect the results.
I'm sorry, could you repeat the question because the sound was not good?
How sensitive are the results of the Bayesian ridge regression to the assumed distribution, which I think is currently normal, and the variance in the priors?
I mean, dealing with a very large number of predictors, it's not going to be that sensitive. I mean, to be honest, I mean, unless we consider a trait that is affected by very few genes. I don't know if that answers the question.
Partly, it's. I'm only thinking that in some of these plants, actually, the assumption of normality, if you took multiple samples, I guess you could rely on the central limit theorem, but some of the, yeah, when you've got a number of markers that tend to be inherited together, you don't have independence in your genomic information. And so you could quite heavily disrupt not just the variance estimates, but the normality, and you could land up skewing it. And I was just wondering whether that was liable to be something you needed to be cognizant of with different plant species.
I mean, yeah, I understand that, but in this case, I mean, it's very clearly our objective is we are not focused on conducting the estimation, right? We are more focused on conducting the prediction, and the way how we evaluate this in general is correlation. So it's not the optimum way to do it, because actually breeders, they don't care about whatever happened in the ballpark or the distribution in the center, as long they are able to select, to identify the most promising genotypes. In any case, we tested or we contrast in general this method versus the conventional method where main effects where we consider, for example, different type of priors for the marker effects. And still the method of interaction was doing better.
Okay, Bulo, you have another question. You want to go ahead? Sorry if I say the name properly. Bulo Tipisa. Okay. He asking about, would you please differentiate the difference between sparse testing and partially replicate design?
Oh, the periods. I mean, I don't think there is a big difference in the way how both concepts are being considered, because in both cases we have used, the idea is just to observe one portion of the data, right, on the field. So here, one of the difference might be that we don't have checks. So we don't, I mean, or the checks are those that we are generating during the different combinations. So the allocation of common genotypes across environments, unique genotypes across environments. Probably those might be a particular case of the sparse testing designs.
I see. Eduardo have two questions. Do you want to unmute yourself, Eduardo? Okay. So the first question that he have is how much the environmental model changes when you move to a crop grow model?
I mean, that's a great question, and I think these are different ways to analyze the data. So they are not like, it depends at the end of the amount of data you might have with grow-up model. Although you are learning most about the physiology of the crop, or you can be probably more accurate, the amount of data that you need to collect is way more. You need to collect data at different stages during the growing cycle, at different locations. You have to focus on a set of genotypes, and then you want to make extrapolations. So probably those might be more accurate. I'm not very familiar with, I mean, I have collaborators here, they're in Florida. I'm not very familiar with the comparisons.
Okay.
The second data.
I see. The second question is, are the plots saying that it's better to have fewer genotype across instead of more genotype across?
I think what it is something that we have discussed a lot. I think what they
he also mentioned that he also mentioned the results not depend on the population structure.
Yeah, I mean, yeah, in general, what the results are telling us is the best is to observe genotypes at least once somewhere, because that is bringing a lot of information to explain the performance of those genotypes in other environments. So no question about that. So when it's about allocation of the resources, the ideal would be to observe probably more genotypes, although those are observed very few times in more environments, because with the genomic data, it's going to be easy just to make up the values of those unobserved combinations.
Okay. So another question, Sanjeev, you want to ask yourself?
Yeah, thanks. Yeah, partly some of the questions are already answered. Yeah. First of all, thanks for a very nice and very informative talk. My first question is, how sensitive is your sparse design to the number of overlapping lines? Like, is there a minimum number which will suffice for, say, 500, 600 lines for sparse design? Or, or how you arrive at a number which is sufficient number for overlapping lines?
Yeah, I think it's, I mean, obviously we have a lot of things coming into the picture, like a population structure or oversampling environment, but assuming that we have enough environments being different and population structure being admixed and having representation of all of these, my feeling and based on the data that we analyzed with from the company, in we don't need more than, I don't know, 1,000 phenotypic records or 2,000. Right? Probably the thing that we have been working lately is 20% should be more than enough to predict the remaining 80% of the genome. I mean, we saw with the company, we have only 2% to predict the remaining 90%. When you have access to these large data sets, probably keep increasing the number of phenotypic records is not going to matter. Probably we increase to 5%. We are not going to go beyond from 0.6 to 0.65. So I think that what we are learning of this is that yes, we can save a lot of resources.
Yeah, my question was more towards the number of overlapping lines in this sparse design.
Yes. So with respect to that, again, it depends on the size of the population, but we observe that in general, no more than 2-3% of overlapping genotypes will be enough to.
Yeah, so that's really good. Thanks. Yeah, and can I ask more question?
Please go fast. We are running out of time.
Yeah. Okay. So you presented result for I think for 4 crops, and then how they were affected by the different models for genomic predictions. Is there also any metric that for each crop, if the validation panel or the test panel was more genetically different than the test panel? Because all 4 crops will not be equally like different between the test panel and the training panel. So is there any metric to test which model was working when the validation panel was very highly different than the training panel?
Not really, because as you said, we assume that all these are coming from the, say population. So it's a little more difficult to think to do this extrapolation outside that. That's why we are considering more this for allocation rather than prediction of genotypes that are not represented in the population.
Okay. Last very quick question. The last 2 slides you presented on Chido, if I am pronouncing it correctly, is it suitable for polyploids or only for diploids?
I mean, so far it's used for. We are not considering the polyploids yet, but if you find a way to have the codification running between 0 and 2, will be more than enough. I mean, it will be useful for the using Chido for run analysis.
Okay, thank you. Yes, that's all.
Okay. There is 2 more questions in the chat. I don't know. I mean, Ishika, are you online? Do you want to read it? Well, the question is Bloop method using in figuring out superior genotype in sunflowers?
Hi, yes, actually, I asked this question. Yeah. So basically, my research is currently on using the blurbs indexes to figure out the various superior genotypes in sunflower. Right. So I just wanted to ask that, is it really a helpful method? Because till yet it hasn't been used in sunflower. It has been used in maize or different crops, but not in sunflower.
I mean, to be honest, I think I have some experience, I think, working with sunflower, but there are many factors that come into the picture, like the population structure, depends on how the genotypes were generated. I mean, blind is difficult to say yes or not. In general, this block has been used for most of the crops. It might be the case. It's going to be quite similar to the case of sunflower.
Okay. Okay. Thank you.
There is a final question. I mean, let me if I can see it. Ian Hunt, do you want to unmute yourself? I don't know, Diego. Can you read the? It's a long test.
CV methods appears to risk leaving a lot of dependence between what is in sample used for estimation and what's out of sample being predicted. That counters the rationale of cross-validation. The most problematic dependence will be between environments and between lines. So you still risk overfitting after CV, which storm in a risk bias towards more complex interaction models. I mean, yeah, this is, yes?
Go ahead. Go ahead, Diego, with the questions.
Okay. Yeah, this is interesting question. As I said before, in general, we're not interested in estimations. There are other type of traits where actually it's very important to be really accurate, like time-related traits, flowering, maturity, etc., etc. In this case, we care about the rankings. And yes, it might be a risk, but in general, the models that include interactions, for example, are being more precise than those models that include main effects. So there is not a perfect model, I would say, that they can deal with all of these issues.
Okay. Is Chido supporting infrared data?
Yes, yes. Yes, actually, that the idea behind Chido is that you can integrate data from multiple data types. It could be nears, the wavelengths, obviously, any data that you can put in a tabular format and you can associate with the phenotypes or genotypes or environments, it can be included in the model.
Okay, but I believe that solves the question. I mean, we are running outside all the time. So thanks, Diego. Thank you very much for taking your time to be here today to give us a nice talk. Thanks you all for being here. So the last thing I wanted to say is like the next start will be that we will have for in the seminar series will be from Paul Gautier from the Queensland Alliance for Agriculture and Food Innovation. That will be in September 11. So I hope to see you, most of you again here. Thank you very much, Diego, again. Thank you very much to everyone. See you next time.
Bye-bye.